In this presentation, I'm going to go through some of the tricky bits about chromosomes and what happens to chromosomes when the cell divides either by mitosis or meiosis, and hopefully that will give you confidence in trying some of the trickier questions. Just a quick recap. DNA is the genetic material of the cell. DNA is a sequence of bases coded as A, G, C and T. Sequences of DNA that code for proteins are called genes. DNA is found in the cell as long linear threads known as chromosomes. And because DNA is such a big molecule, the DNA is wrapped around histone proteins which compact it and condense it so we can fit it into the nucleus. This shows all the chromosomes in a normal human body cell. It is a diploid cell. Diploid means that there are two of each different chromosome. You can see here there are two copies of chromosome 1, one there and one there. There are two copies of chromosome 2, one there and one there, two copies of chromosome 3. These copies are known as homologous pairs. The N number of a cell is the number of different chromosomes. So a diploid cell has 2N chromosomes. For a human cell, N is 23. So a diploid human cell will have 2 times 23, which equals 46 chromosomes. This shows a homologous pair of chromosomes. The homologous pairs are the same size and shape, and they contain the same genes at the same locus, although these genes may be alleles or variations of genes. Now, before the cell can divide, the DNA must replicate. This takes place in interphase. You can see now that our homologous pair of chromosomes now exists. Each chromosome is now found as a pair of sister chromatids held together at the centromere. These are genetically identical because the chromosome has just replicated itself. Although the number of chromosomes has stayed the same, we've still got 2 chromosomes, but the amount of DNA has doubled. This shows all the chromosomes from a human cell. So it's similar to the picture that I showed you earlier, but now you can see that each homologous pair has now replicated. So the chromosomes are now seen as sister chromatids. So this is our homologous pair, 2 chromosomes, but each existing as sister chromatids. Mitosis is cell division that produces 2 genetically identical daughter cells. It's important to make new genetically identical cells for growth of the organism, repair of tissues or replacement of cells. An additional mark point could be to produce cells that can differentiate to form tissues or for asexual reproduction of some organisms. If we look at these drawings of cells taken from different stages of mitosis, we can see that there's a logical sequence to. So we start off in late interphase, early prophase. The DNA will have replicated at this stage, but you can see the nucleus is still intact and the chromosomes are found as long, thin threads. The cell, as it goes into prophase, here the chromosomes start to condense and the nuclear membrane starts to break down and spindle fibres start to form and they will attach to the centromeres. The spindle fibres will then help move the chromosomes to the middle or equator of the cell. This is metaphase. The spindle fibres then contract and the centromere splits. So now the chromatids come apart and start to move towards the poles of the cell. When they finally get to the poles of the cell, this is telophase, and then the cytoplasm will start to form inwards and divide, forming 2 new genetically identical cells. The nuclear membrane will reform to form 2 new nuclei. This is telophase. Once the chromatids have separated when the centromeres split, they are once again known as chromosomes. So we can see that at the end of mitosis, the new cell here will also have a diploid number of chromosomes. It will have exactly the same number of chromosomes as the parent cell, and the amount of DNA in the cell has gone back to normal. So the DNA has now halved and the chromosome number has stayed the same. If it was a 2N cell or a diploid cell at the start, it is again a diploid cell at the end. So meiosis is a different type of cell division. This time we get 4 genetically different daughter cells with only half the number of chromosomes, one of each homologous pair. These cells are called haploid cells. Reasons for meiosis. First one, to halve the chromosome number so that we can restore the chromosome number when the gametes join together at fertilisation and to enable genetic variation. This shows all the chromosomes in a human haploid cell. You can see that now there is only one copy of chromosome 1. It's a haploid cell, so now it has 23 chromosomes, n equals 23. So let's quickly look at meiosis. Here we've got a diploid cell. It's got 2 homologous pairs of chromosomes. It's at the start of interphase, so the number of units of DNA is 2. Before the cell can divide, the chromosomes replicate themselves, and we can see here the chromosomes are now seen as sister chromatids joined at the centromere. The amount of DNA has doubled, so now we have 4 units of DNA. Now, meiosis has an extra cell division. At first, we've got a reduction division. So what happens is that the homologous pairs find their partner, and the pairs line up on the equator of the cell in metaphase 1. The spindle fibres will then contract and pull the pairs apart. So when the cell divides here, we will now get 2 haploid cells, here and here. The amount of DNA in the cell has now halved. The cell then goes into a second meiosis type division, which is essentially the same as mitosis. You can see that the chromosomes are lined up on the equator attached by spindle fibres in metaphase. The centromeres split and the chromatids move apart, and we end up with 4 haploid cells with just 1 unit of DNA. Let's quickly look at how meiosis causes genetic variation. There's 2 ways: crossing over and independent segregation. Independent segregation, here we've got 2 homologous pairs of chromosomes. This time I've just drawn them slightly different colours so that you can visualise the two pairs. You can see that when the pairs separate during meiosis, these 2 will lead to one cell and these 2 will end up in the other cell. However, if they line up in a different orientation, when they separate, we'll get different cells forming. This leads to genetically different gametes. To show you crossing over, I've gone back to the original cell with the 2 homologous pairs. In crossing over, the pairs, when they line up in prophase and metaphase, they can get tangled up. And when they get tangled up, they break off bits and they swap bits of genetic information. So you can see here that a little bit of the orange chromosome has been swapped with a little bit of the red chromosome here. This means that now when the chromosomes separate to halve the chromosome number, we can get a real mix. We can have black with a red. Here we've got a black with a bit of orange and red, and again different gametes forming. Here are key mark points if you were asked a question about genetic variation and meiosis. Okay, here's a quick exam question. The exam question tells me that this, figure 3, shows the chromosomes in a diploid cell. I know that diploid means that the chromosomes must be in homologous pairs. I look at this cell and I can see I've got 2 long chromosomes and 2 short chromosomes. The 2 long ones must be the pair because I know that homologous pairs are the same size and shape. This must be the other pair. I also know that when the cell divides, and this is the important bit, that the gametes must only have one of each pair. So this shows the possible combinations. When they pair up, only one of each pair must go to the gamete. So I can have a big white one with a little white one. I can have a big white one with a little black one. I can have a big black one with a little black, or a big black with a little white. They're the only combinations. Okay, let's look at some of these questions where they ask you to identify what's going on in the cells. When I'm looking at these, first of all, I'm looking for homologous pairs, and I'm looking for whether the chromosomes have replicated or not. I can see straight away that cell E is a diploid cell because we've got 2 pairs of homologous chromosomes. Remember, the homologous pairs are the same size. However, these chromosomes have not replicated, therefore this must be at the start of interphase. We can see that B also has 4 chromosomes in 2 pairs, so it's a diploid cell, but here the chromosomes have replicated. So this must be at the end of interphase or when it's going into cell division. D is also a diploid cell. Over here we've only got 1 pair, but these are a pair of homologous chromosomes because they're the same size. A and C are both haploid cells. We know they're haploid because their chromosomes are different sizes, therefore they are not homologous pairs. A these chromosomes have not replicated, whereas in C they have replicated. We can now fill in the answers. So a haploid cell at the start of interphase, that must be A, and a haploid cell in prophase must be C. A diploid cell at the start of interphase, that must be cell E. A diploid cell in prophase, where the DNA is replicated, that must be B. 2 N chromosomes where N equals 1, that is D, and 2 N chromosomes where N equals 2, that could be both B or E. Just a quick summary about the life cycle of an organism. We start off with 2 haploid cells with N number of chromosomes. These gametes will come together in fertilisation. To produce a zygote with 2 N chromosomes. That zygote will then divide by mitosis, leading to growth and a 2N adult, which will then produce gametes by meiosis. Here's a question based on life cycles. Now don't panic, you really don't need to know about the life cycle of a moss plant. The clues will be in the question. Here we've got an embryo, it tells us it's 2N, that means it's diploid. Here this plant is still 2N, diploid. This must have divided by mitosis. Here we get a spore, which it says it's N, that tells me it's haploid. So that tells me that from here to here, meiosis must have taken place. On the diagram, mark with an M where meiosis takes place. M. Here's another question. Basically, again, the clues are in the question. We've got stage A, and we've got 4 chromosomes here, and stage B, we've only got 2 chromosomes. It says, what's the diploid number of chromosomes in this fern plant? Well, I know that a diploid cell has twice the number of chromosomes as a haploid cell, so therefore that tells me that the diploid number must be 4 chromosomes. The question then asks me to explain the difference in the number of chromosomes at stages A and B. Well, again, from looking at the question, I know that meiosis must have taken place, therefore the number of chromosomes has halved. Just to finish, make sure you know the keywords and that you can answer these questions.